Hi, everyone. Welcome to Spatial Omics Seminar Series 5 again. And today we are very happy to have Dr. Omer Bayraktar from Wellcome Sanger Institute, UK. And briefly, he received his Bachelor's of Science in Molecular Biology and Genetics from Middle East Technical University in Ankara, Turkey. Then he received his PhD in Biology, Institute of Molecular Biology from University of Oregon in the US. Then after that, after a postdoc at University of California, San Francisco for about 5 years in the Department of Pediatrics, Dr. Bayraktar moved to University of Cambridge. And after a year of postdoctoral employment, he started his own lab as a group leader in 2018 in the Wellcome Sanger Institute. And today he will be showing cool spatial transcriptomics technology integrating, I think, RNAsea technology as far as we know, and Nanostring, and maybe more that he may have in his own lab. And without further ado, then I'd like to turn to Dr. Bayraktar. So
lovely. Thank you, Ahmet, for the lovely introduction, and both you, Ahmet, and Rong for inviting me. It's feels like a really nice sort of like informal chats type of situation, which I enjoy. So it's really a pleasure to be joining all of you today. You know, sort of like Ahmet led, I'll be telling you about the work we're doing in my lab at the Sanger Institute on really combining different types of omics methods to start, you know, creating sort of highly detailed spatial maps of complex tissues. So, you know, really, I think to keep up with this, you know, to keep up with the sort of informal nature of it, you know, if there's something that you're really like not clear about, please feel free to interrupt, and I'll be happy to take questions during the talk as well. So today, what I'll tell you about mostly is that how we can combine really single cell genomic methods with various types of spatial genomics to really start, you know, dissecting tissue architecture at scale. So I think, you know, this crowd really needs a very little introduction to any of these technologies. I think most of you are very well aware that, you know, single cell RNA-seq, while it's a fantastic way to sort of get a molecular catalog of very different sort of cell types in a given tissue is based on unbiased methods, it actually loses the spatial context of the cells, right? To be able to really understand how all these different cell types are putting together complex tissues or how they are talking to each other in disease, you know, for studying adherent organs, we really need to be able to put all of them back into their tissue context. And to be able to do this, you know, over the past couple of years has been this incredible explosion of different kinds of spatial genomics methods. And I think we could sort of roughly bin them into the methods that are either trying to sequence stuff in tissue, or the methods that are, you know, more based on imaging that are trying to, you know, use cyclic multiplexing methods. Approaches to really, you know, interrogate the expression of very many RNAs and proteins. You know, in this sort of big picture, the part that we are really interested in is, you know, developing both experimental as well as computational workflows that will allow us to put all these different technologies together to come up with multimodal representations of tissue. So along with that, what I'll do today is that I'll start by telling you guys about the tools that we've started to develop, the computational tools we've started to develop to put different kinds of spatial omics data together with single cell RNA-seq. And once I'm done with that, I'll tell you a bit more about some of the new spatial transcriptomic technologies that we've been trying out in the lab. So I'll start my sort of, I'll start this by talking a bit more about spatially resolved sequencing than imaging. And, you know, I'm doing that because, you know, spatially resolved sequencing methods, they're like actually quite closely related to single cell RNA-seq. You could really consider them as cousins, right? They're both methods that provide you unbiased transcriptome-wide analysis. A lot of the spatial RNA-seq methods are tend to be sort of like simple in their execution. So, you know, the sort of three of the many methods that I decided to, three of the many available methods that I decided to show here, you know, they work on sort of like simple histology molecular biology protocols where you start with a thin tissue section, you permeabilize it to capture, you know, transcripts onto a positional oligonucleotide array. And once you have this transcript capture, you can just read them out using NGS. In this field, I think one of the most exciting things has been the sort of the rapid increase in the resolution. When the technology of spatial transcriptomics came out several years ago, you're looking at these 100 micron spots on the spatial arrays, which corresponded to over a dozen cells in many tissues. At the moment, I think the sort of the most commercially available spatial omics technology, Visium, is giving you this 50 micron resolution, which depending on the tissue gives you something like 2 to 5 cells. And you're probably all aware of higher resolution technologies ranging from slide-seq to dbit-seq from Rong, or to other crazy methods like the BGL nanoball-seq that give you much higher spatial resolution. So these data types are great, but from these types of spatial transcriptomic methods, the data that we get from them is not quite equal to single-cell RNA-seq. And the simple reason for that is that in all of these different technologies, we are actually doing a grid type of profiling of the tissues, so that there isn't a one-to-one correspondence between a given cell in a tissue and their array position. This is quite obvious with technologies like Visium, where you know that the size of these spots is much larger than one cell and contains multiple cells. But even with higher resolution methods like slide-seq, now we know that each of these sort of puck or spatial positions receive mRNA from fractions of multiple cells. So to really truly analyze these data types like single-cell RNA-seq, we need to be able to computationally deconvolve cell type specific information from these data. So that's precisely the area that I want to talk to you about. So, to be able to do this with diverse spatial transcriptomic data types, we recently teamed up with Oli Stegel from DFKC and EMBL, and under the sort of great leading of our graduate student Vitaly Kleshchevnikov, we developed a probabilistic model that we call cell-to-location to integrate multiple different kinds of single-cell and spatial transcriptomic data. The sort of simplified approach is shown here. We start cell-to-location with a single cell and spatial transcriptomic data set from your tissue of interest. What matters the most is that these data sets match each other as much as possible, such that they are from the same sort of tissue region and organ. I'll show you workflows where we can actually generate these data sets from the same organ, but you can certainly use single cell data sets that have been generated externally to spatial transcriptomics. What cell-to-location does is that it uses a Bayesian probabilistic framework to actually define the cell type expression signatures of your cells of interest from the single cell data and deconvolve the spatial transcriptomic data set to estimate the abundance of all of these reference cell types in the tissue. And once you have this, you can use that for more fun applications, such as automatically identifying tissue regions based on their cellular composition, or doing cell co-location analysis to identify, co-localized and putatively interacting cell types. So today I'll just give you a very high-level description of the model. In our workflow, we are starting by deriving gene expression signatures of our reference cell types from the single cell data. If you want, you can feed sort of like your favorite method or sort of conventional method of use to just estimate the average gene expression profiles of each cluster, but we also provide more specialized negative binomial regression models to actually extract these gene expression signatures, dealing with things like batch effects. Then in terms of spatial transcriptomics, our workflow is really applicable to any count-based data. I'll show you examples of all three of these methods here, but really, you can use any of them. And then cell-to-location is basically using the reference gene expression signatures to estimate both the absolute as well as relative abundance of each of these cell types at each spatial tissue location. And that's what's actually giving you these inferred cell type positions. So under the hood, cell location is a negative binomial of counts in spatial transcriptomic data. The sort of most important aspects of the model, I think, that define it, that distinguish it from a couple of other similar methods, is that first, we are taking into account the platform effects. We are actually modeling the sensitivity differences between the single cell and different spatial transcriptomic technologies. We are also taking into account things like additional background signals in spatial transcriptomic data, such as free-floating DNA, etc. And on top of it, we are also using the co-location of different cell types as a prior. So this is basically making use of the fact that in most tissues, functionally related cell types tend to be next to each other in location. And we're actually using this cell co-location as a prior to increase our accuracy. So those of you who are familiar with this kind of computational deconvolution know that there's a number of great tools that came out over the past 6 months. With cell location, what really sets us apart is that we've really specialized to deal with complex tissues as accurately as possible. It allows you the scalability to look at very complex single cell references that may contain hundreds of cell types. We think, as I'll show you with some of our benchmarking data, we think we're better at mapping rare cells and transcriptionally subtle transcriptional states, cellular states. And we're versatile across spatial technologies. So, based on our hierarchical model structure, it's really easy to switch back and forth between, say, Visium and Nanostring data. And finally, we're computationally efficient, so we can scale up. So when we developed cell location, we wanted to have like a really nicely characterized single cell and spatial data sets that we can use to actually test drive our spatial mapping. So to do this, we actually generated a new single nuclei, a new paired single nuclei and Visium spatial transcriptomic data set in the mouse brain. And what we did is diagrammed over here. So, in a particular region of the mouse brain, we derived a number of thick and thin consecutive tissue sections. The thick tissue sections, we use them to extract nuclei and generate a 10x V3 unbiased single nuclei reference. And the thin tissue sections, we place them on the Visium arrays to generate, these spatial transcriptomic data at the 55 micron resolution. And then we use cell location to put them together. We picked a region of the mouse brain as shown here with the histology image of the very tissue section analyzed. The histology image of one of the very tissue sections analyzed with Visium. We picked an anatomical level that contains many brain regions. So we know that there are very many different cell types that are expected to be localized to these different brain areas. And with our spatial data, with both of our single nuclei and spatial data, they're high quality data sets that give you good transcriptomic depth. So here I'll jump right into our results. On the left, what you can see are single nuclei reference, where we have done kind of a medium depth type of annotation to identify 59 different transcriptomically distinct neuronal and glial cell types. On the right, I'll play you a little animation where I'll show you how we take 6 of these cell types and map them into their known locations in the Visium spatial transcriptomic data. So these 6 cell types here are all sort of, well-known neuronal and glial cell types that are known to be localized to different parts of the mouse brain. Throughout the animation, you'll see how we calculate their average gene expression profile and map their abundance across different Visium spots on this very same tissue section, where you can see the histology image on the background. So I love showing, this kind of gimmicky animations to really demonstrate how we're linking the single cell with the spatial data. But really, I think the main advantage of doing this type of work is that we are not just mapping 6 cell types here. We're taking all of the cell types in our reference and inferring their locations in the spatial data set at once. And I think what that does is that it really opens the door to doing, high throughput and fully automated tissue mapping. So here, what we've tried to do is to sort of show you some of the fruits of this kind of mapping. So on the left, you can see the histology image of the tissue section where we have annotated with the different brain regions, just based on manual annotation. In the middle, what we've done is that we have actually clustered each of the sort of visium spots based on the estimated abundance of each of the 59 cell types from cell to location. And when we do this, we can actually identify in an automated manner the tissue regions present on this tissue section. And if you're familiar with the mouse brain, neuroanatomy, you'll notice that really a lot of the sort of different areas that we have found here, ranging from cortical layers to hippocampal divisions to different parts of the diencephalon, nicely match what's already known. And with these automatically identified tissue regions and automatically mapped cell type locations in hand, you can really start generating these types of tissue maps in scale, right? So the dot plot on the right, you can see that we really identified all these different brain regions in a highly automated manner, and we could really come up with detailed maps representing both sort of highly regionalized cell types, such as very specific neuronal cell types, as well as more ubiquitous found cell types such as glia between all these different brain regions. So to showcase our strengths here, the strengths of the cell to location model, I'll just focus on one set of results from the mouse brain. And that is our ability to really map and distinguish transcriptionally fine cell type populations. So the population that we're focusing on here today are astrocytes. So astrocytes are a very, important and, regionally ubiquitous glial cell type in the nervous system. They are sort of a special type of cell to me. As I spent a good deal of my postdoc trying to understand if astrocytes in different parts of the brain were molecularly and functionally distinct. So I was really, when we generated this data set, I was quite interested to see, can we actually find any kind of new regional astrocyte heterogeneity? So what we did here is that we basically pulled out all the astrocytes from our single nuclei data and performed subclustering to identify if we can find more subtypes. And when we did this, we identified about 10 molecularly distinct subtypes that we could distinguish based on the expression of different markers. But we really noted that the transcriptional differences between these subtypes were not that big. They were distinguished by the expression of a few dozen genes, while if you did the same type of analysis for neurons, they would be distinguished by the expression of hundreds of genes. So we didn't quite know what to make of these subtypes until we actually mapped them using cell to location to the visium data. And when we did that, it was very obvious that these subtypes represented astrocytes from different brain regions. So in this little map here, you can see astrocytes that we found to be very specific to the cortex versus the hippocampus versus the thalamus and the hypothalamus. Some of these subtypes, such as the cortical subtypes, were known, but a bunch of the others were actually new to neuroscience as far as our knowledge. Amongst these, I'll actually focus on what we think are the newest type of astrocytes in the field. And those are the ones that localized to these brain regions called the diencephalon, specifically the hypothalamus and the thalamus. So what you can see here on the top are basically the cell to location regional spatial estimates of each of these cell types in the visium data. You can see that the subtype that we annotate as the hypothalamic is highly enriched in there, while in the thalamus, we have identified 3 distinct populations. The most surprising was this guy over here, which is a very rare astrocyte subtype that we found to be very specifically localized to the medial habenula, an interesting nucleus of the thalamus involved in behaviors like addiction. But then we also identified subtypes of astrocytes that distinguish different parts of the brain, namely the medial and the thalamic, medial and the lateral thalamic divisions. And these estimates we could nicely validate them using orthogonal RNAscope-based methods. So what you're seeing here is now quantified RNAscope single molecular fish data at a single astrocyte resolution, where we basically multiplexed markers of these different subtypes with markers of astrocytes, and then counted the expression, quantified the expression level of relevant markers, and the patterns that we got from this SM fish validation really nicely matched the subtle location estimates. So the population that I really like to bring your attention to here is this little interesting astrocyte subtype that we mapped to habenula, and I think it's a really nice poster child for self-location, because first off, it's extremely rare in our single nuclei data set. It makes up about a little less than 0.1%. In terms of its spatial location, it is very highly enriched to this really small brain region. So it's really represented by a few locations. And as you can see with an orthogonal method, it really nicely maps out. Its specificity is really to this brain region. So I think it really nicely shows that, with this kind of method, even if you're working with high spatial resolution Visium data, you can really take, highly specialized transcriptionally fine populations and map them here. And while our benchmarking efforts to really identify the advantages of self-location are ongoing, it's becoming clear to us that we are indeed better than most of the other sort of spatial transcriptomic deconvolution methods in dealing with these fine-grained and rare cell types. So what you're seeing here is now repeating our spatial mapping exercise in the mouse brain using multiple different methods, including RCTD, SCVI stereoscope, and SIRAD V3. And you can see that just looking at this habenular astrocyte subtype, self-location is really the only one that maps it to this medial habenular brain region with higher confidence. And we see that really as we look into sort of other tissues and other fine mouse brain cell types, our benefit is, our sort of performance benefits are also replicated. As I wrap up the mouse brain part, I'd like to also show you that, with self-location, it's applicable to diverse tissue architectures. So, the mouse brain, well, it's very close to my heart, and it's this beautiful organ for spatial transcriptomic mapping. It's also relatively sort of an easy tissue on the different spectrum of tissues. And the main reason for that is that, it shows high regionalization. That in different parts of the mouse brain, there are very representative dominant regional cell types, such as cortical layer cell types or thalamic cell types. But many other tissues in our bodies are quite different. In most of the immune organs, such as the lymph nodes, for example, what you have is that you have highly spatially intermixed cell types, and these could often be, closely related cell states, such as, different flavors of T cells and B cells. So we were quite interested to see how self-location would fare in a tissue like the lymph node, and we basically repeated a very similar exercise. We took a sort of an integrated, single-cell RNA-seq reference of secondary lymphoid organs, and we mapped it to a publicly available Visium data set of the human lymph nodes. When we did this, we passed the first test really easily. We could identify kind of the major lymph node divisions, such as the germinal centers, T cell zones, or B follicles that would be distinguished by these canonical populations. But we could go beyond that and actually start to distinguish the spatial positions of much finer immune cell states. So what I'm showing here is a slightly different version of the tissue region analysis. So what we've done here is that we took all of our immune single cell subtypes, which also included some stromal types. We mapped them to the lymph nodes with self-location. Then we actually ran non-negative matrix factorization to identify cell types that are more likely to be located, co-located with each other, right? And this kind of analysis spat out, these factors that contained, immune and non-immune cell types that are actually known to be localized to different parts of the tissue that are known to interact with each other. So with this, we could identify fine divisions such as, B and T cell states that distinguish dark versus light zones or the germinal centers, which is a very important sort of spatial switch in B cell biology. And really, looking at this, we could identify really fine flavors of each of these immune cell types and locate them to different bits. And again, as I told you before, with the mouse brain astrocyte subtypes, I think with the lymph nodes, we are also better than other methods in sort of doing mapping of these fine cell type populations. So what I'm showing here is just one T cell subtype, a specialized follicular helper cell type, or the germinal centers. This is a cell based on prior knowledge that you expect to be very specific to the germinal centers. With cell to location, that's indeed where you see it. With the other methods, a bunch of them indeed do manage to map it to the germinal centers, but you often see a lot of noise. You see that they actually tend to map it to many other non-germinal center locations of the lymph nodes as well, showing you that, with this fine cell type, they're struggling a bit in terms of its very specific and accurate mapping. And we think, these types of advantages in mapping challenging cell populations would be quite important moving forward in lots of different tissue atlasing projects. I think in lots of sort of single cell and spatial transcriptomic profiling projects, often the most interesting cell types are those that are either sort of like fine cell populations or those that are rare. Right? Because to be honest with you, I think even in most human tissues, we already know which cell types are kind of the major dominant ones, right? So we've been quite happy, I think, over the past, several months to, collaborate with a number of folks on applying cell to location to different tissues. For example, I'm showing you here a lovely preprint that we got to contribute to from the Teichmann lab, where we actually performed spatial mapping and cell to location on Visium spatial data, spatial transcriptomic data of the human fetal gut, where we could identify both sort of like these major, gut cell types, such as enterocytes versus, panel cells and stem cells, but we could also start mapping sort of fine, immune cell populations, such as those that constitute the Peyer's patches with cell to location. Before I wrap up the cell to location part, I would like to give you a sense of sort of the different directions that we're going in here. So one direction that we are quite interested in investing is basically aiding the prediction of cell-cell interactions from single-cell RNA-seq data. So I'm quite sure that, a lot of you are familiar with, sort of cell interaction analyses from single-cell RNA-seq data. There are now a number of wonderful tools like CellPhoneDB that sort of, use, curated lists of receptors and ligands that bind to each other to look at single-cell data and in a sort of permutational manner, actually predict which cell types are likely to interact with each other based on the expression of these extracellular proteins. So, you can nicely generate these kinds of like plots, predicting which cell types would interact with each other. But you'd appreciate that in most solid tissues, a problem we might have is that some of these cell types that express matching receptor and ligands might not be closely located to each other. They might not be located in the same part of tissue to be engaged in short-range interactions. So we've been quite interested in basically using our spatial mapping method to aid the identification of cell types that are near each other that we can use to basically refine cell interaction analyses. And we were happy to be able to do this as part of a fantastic collaboration with Rosa Tormo and Sarah Teichmann, our colleagues here, in mapping the human endometrium, where they generated a wonderful sort of single nuclei and Visium survey of the the uterine tissue, and we could actually come in here with cell to location, map very different cell shapes, such as these new epithelial cell types that they identified. But then we could actually use this co-located cell type information to be able to look into CellPhoneDB analysis of single cell data and only focus on the interactions between cell types that are known to be closely located to each other in the tissue. Hence, we think that there's lots of, I think, potential in coupling spatial transcriptomics to cell interaction analyses. And finally, as I'll switch to in the second part of the talk, we're actually quite interested in applying cell to location to different spatial transcriptomic technologies. And what we've been able to do here is to really take the, the slide-seq V2 data of the mouse brain that was generated by Ahmet Coskun and Fei Chen, and take our single nuclei reference of the mouse brain and map it. When we did this, we could sort of nicely find these hippocampal as well as cortical layer populations, showing that our method is also applicable to high spatial resolution transcriptomic data. So with that, I think I'd like to switch gears a little bit. I hope I convinced you that cell to location allows you to sort of really combine spatial and single cell transcriptomics in a number of different sort of tissues, in a number of different sort of tissue structure contexts, and really build these spatially heterogeneous tissue atlases. In addition to our computational work here, we're also interested in, contributing to this ever-expanding sort of toolkit of experimental methods. And, with Visium, I think it's really nice commercially available methods. It's been, I think, fun to apply to different tissues, requires a little infrastructure. But I think, there is basically lots of room for different kinds of technologies that address different types of questions. And one of them that we've been looking at is the Nanostring whole transcriptome atlasing method, or WTA for short. And I think, in this table, what I try to communicate is that this method, while it also provides transcriptome-wide information, what it really excels is in deriving cell type specific or targeted sort of profiling on archival FFPE tissue material. That's the application that it's really that really distinguishes it. So how does it work? So I think a lot of you are familiar with Nanostring, so it will not be, and you've probably heard about, I think WTA from Joe Beecham or one of the other Nanostring folks. But this is a reminder what a WTA method is. It's basically a hybrid between in situ hybridization and sequencing. So you basically start with a sort of a standard tissue section, like an FFPE section on a microscope slide that you mark using either a Sunfish or IHC for a small number of cell type markers or tissue markers just to get your bearings. After that, you perform in situ hybridization with the special WTA probe mix that contains 18,000 probes targeting effectively, I think the full human transcriptome. So these probes operate based on the sort of the Nanostring geomics technology. They have an NGS. In their little sort of they have their target specific hybridization region, but then they have this little NGS tag tail, and that tail is separated from the main probe by a photocleavable marker. So after the hybridization, the slides go into the Nanostring GeoMx DSP machine, where you can perform really spatially precise photo elimination, aided by the the imaging of the markers that you put on in the first place to either target select tissue regions or specific cell types. And with this photo cleaving event, you end up basically removing these NGS barcodes from each of these tissues. Then you suck them up from each sort of illuminated region of interest, or ROI, using a little sipper, put it into a plate, and read them out using sequencing. So, when this technique came about and then we started our collaboration with Nanostring, we were quite interested to see if we could apply it to neuroscience. And we thought that it would be a really good match to study the development of the human cerebral cortex, which is a piece of tissue that shows a high degree of regional organization. So I think, we could talk a lot about the spatial architecture and the different cell types of the human cortex, but for the purposes of this talk, what is, I think, most critical is knowing that in the cerebral cortex, you have sort of like these two main regions. The sort of the deep part of the cortex includes the germinal centers, which contains basically different types of neural stem cells and progenitors that undergo asymmetric as well as self proliferative as well as asymmetric self-divisions to give rise to differentiating in neurons and glia throughout embryonic development and fetal development. These differentiating neurons and glia then actually radially migrate out to the more superficial parts of the cortex, namely the cortical plate, to give rise to basically the sort of the maturing 6 layers of your cerebral cortex. So, if you were to really sort of spatially profile the different cell type populations across this axis of cortical depth throughout the germinal centers and the cortical plate, you are really capturing the sort of cellular axis of cortical neural differentiation. Wonderful postdoc in the lab, Kenny Roberts, worked with application scientists from Nanostring WTA and Prof to actually apply this to the human brain at mid-gestation. So what you're seeing here in this panel is basically a mid-gestation temporal cortex tissue section where we have performed RNAscope against 3 different markers of different cortical neural progenitor subtypes. Once we did this, we could nicely distinguish these fine neural sort of germinal cell type populations throughout the cortex. And afterwards, we basically performed a bulk segmental spatial profiling experiment where across the cortical depth, we basically drew these white boxes, or ROIs for a more professional nomenclature, these white boxes across cortical depth. Each contained dozens to hundreds of different cells, and then we basically collected all of the different RNA signal from each of these different compartments. The first thing we wanted to see is that at this bulk tissue level, can we actually get transcriptome-wide readout? And the answer to that was actually yes. So what you can see here is the number of transcripts detected in each of these different ROIs across a number of technical and biological replicates. And you can see that, on average, when you have something like a couple hundred nuclei to 1,000, we are indeed detecting the expression of like 8,000 to 10,000 genes. So we are getting a transcriptome-wide readout. While I will not go into great detail on the sensitivity of the method, if you check out the preprint that we put out, you can also see that we can push the application of this down to near single cells. At the single cell resolution, our sensitivity is a little less. We can only read the expression of something like 600 to 1,300 genes, and our throughput is basically low. But I think it basically shows you that, you can actually make this technique work from low input material. Though our recommendation really is that you use it to look at either bulk tissue profiling or profile groups of cells, such as half a dozen or more. So before I show you the cell type mapping, I'd just like to demonstrate that this kind of data set, you can really, start diving into the transcriptome-wide nature and start making conclusions. So for example, one of the things that we were interested in looking at was basically the spatial expression pattern of genes associated with different neurodevelopmental or, psychiatric disorders. And, one area that we were particularly interested in was autism, because previous studies that used approaches like bulk RNA-seq and co-expression analyses, such as this wonderful paper from Matt State and Jeremy Bilsey, they all pointed at deep cortical layers as a convergence at the expression level of many different autism genes. But there was relatively little basically in situ validation data that suggested that this was true. So in our hands, what we could do is that we could basically compile lists of genes, risk genes for autism or different, neurodevelopmental disorders such as, IQ, genes that are linked to brain size, or much more comprehensive lists of neurodevelopmental disorder related genes. And then we could simply ask at our bulk Nanostring WTA data, if we looked up the enrichment of these gene sets, do you actually see that they are enriched in a particular part of the germinal centers on the cortex or the cortical plates. And as you can see here, specifically for the autism genes, we find that their expression was much higher enriched in the subplate and the deep cortical layers, which is demonstrated here relative to others, really showing that we could use this technology to interrogate the expression of very many genes and make use of the transcriptome-wide information. As I showed you in the first part of the talk, we are also quite interested in putting this technology together with single-cell RNA-seq. And another fantastic grad student from the lab, Alexander Ivazidis, what he did was he basically developed a new version of cell-to location that is tuned into more accurately into the sort of background signal of the nanostring assay to actually integrate WTA with single-cell RNA-seq. So what I'm showing here is now Alexander's work, where he took a publicly available single-cell RNA-seq data set of the mid-gestational cortex from the Geschwind lab, which contained a number of different cell types. At one annotation level, you can see sort of the 16 cell types of this single-cell reference that represent different flavors of neural progenitors, neurons, and glia. And we basically deconvolved our bulk nanostring data to each of these cell types at each of these spatial positions. When we did that, we saw this sort of really nice, known mapping of these, cardinal developing brain cell types. For example, we could identify that, different flavors of neural stem cells that are known to be localized to the ventricle versus the outer subventricular zone were nicely distinguished. But we can also find, again, sort of like the more sort of like, challenging cell types. For example, we could identify that newly born neurons, which are transcriptomically very distinct from later born neurons, we could find them in the germinal centers where they are born. Slightly more mature migrating neurons, they could be found in transit between the two areas. And then the different flavors of upper versus deep layer differentiated neurons, we could also map them to different layers of the developing cortical plates. And with something like this, again, I think really the main advantage is that you can, it's a high-throughput technology, so you could really apply it to, different samples, different ages. So when we did this in the same brain region at two different developmental, time points to the cortex, we could really nicely see these spatiotemporal, changes in these cell populations. So for example, from 14 to 19 post-conception weeks, we can nicely see that interneurons, which are made in more ventral parts of the brain and actually migrate into the cortex throughout the germinal zones, how they actually increase in size, but also we could track the accumulation of excitatory neurons in the cortical plate and the migration order of different neuronal subtypes and the refinement of the layers. And finally, I think one of the actually most compelling uses of this WTA technology is that as opposed to grid type of profiling, it allows you to just focus on a couple of different cell types and just profile them instead of the whole tissue. And that's done in a manner that couples image segmentation to photo cleaving. So demonstrated an example here where we are looking at now the germinal zones where with RNAscope we could really mark neural stem cells versus intermediate progenitors, these different neural progenitors in different colors, and we could actually automatically segment this RNA signal to identify the locations of these different populations and actually profile them separately. When we did that, you could see that by doing differential gene expression analyses, we could pull almost over 1,000 genes that are known to be differentially expressed in these cell types, mapping really the sort of the expression pattern of these markers from single cell data, suggesting you that we could really do cell type specific profiling with this technology. So with that, I hope, in sort of both the computational and the experimental bits, what I sort of, what I could show you was that with the right combination of the experimental and computational approaches, I think, the combination, the integration of single-cell spatial RNA sequencing allows you to really map complex tissue architecture and tackle these complex tissue materials. I think in both the Visium and the nanostring examples, one thing we try to demonstrate is that you could actually use this integration to sort of computationally push the resolution of your spatial transcriptomic data sets. So I think, the preprints pertaining to the work that I presented today, as well as both the cell-to-location and nanostring data analysis tools, they're both, online at the moment. So, if you're interested, please free to check them out. And as closing remarks on what we're looking at next on the computational side, the two bits that we're thinking about the most right now is that first, I think as we generate more spatial transcriptomic data sets, we are starting to realize that, a lot of the things that plagued us with single cell data are there again with spatial transcriptomic data. So things like experimental batches between labs or within labs are still there. And I think, the next generation deconvolution methods, they really need to be able to take these into account. Finally, I think a big part of the future of spatial technologies is large-scale applications, be it methods that give you higher spatial resolution, or just the ability to really apply these to very many different tissue sections. So we're really talking about a massive increase in the size of these data sets and their complexity. So we're also quite interested at the moment in tweaking self-allocation to make it play nicer with spatial transcriptomic data sets that have hundreds of thousands to millions of locations. And we're quite excited to be able to share some updates with you guys soon. So with that, I hope that that was clear. I think I'll just finish here by acknowledging the lab, the wonderful set of postdocs and grad students who did the work, as well as our collaborators. Most importantly, Oli Stegel on the self-allocation side, but also 10x Genomics and Nanostring on their help with Visium and WTA analysis. With that, I hope you enjoyed that. I'm very happy to take any questions.
Thank you, Omer, for this great presentation. And any questions? I see Roy, maybe.
Yeah, I can start. So thank you very much, Omer. It was really fantastic. And I completely agree with you that the need to integrate single cell RNA-seq with spatial transcriptomic is definitely there, right? Because they're basically both measuring, in some sense, the same thing. But what amazes me, and really interesting to hear your thoughts about it, is the fact that the reference itself is not maybe fully well-defined, right? So in some sense, why did you guys have to redo the single cell RNA sequencing when the Allen Institute already imaged, millions of cells with the same technology? Yet you did because probably, as you're saying, right, different labs, different hands, it's not the same. So we're trying to use this data, but yet it itself is not really, it's a moving target. Right. So how can you use something as a reference when it's a moving when you're not fully well defined?
Yeah. So that's a great remark, Rowie. And I think my answer to that is basically yes and no. Right. So I think that actually the primary reason that we generated our own single nuclei data set was that we really wanted the perfect match between the single cell and spatial transcriptomic technology. And we thought that we really needed something like this is pretty much as good of a match as you're going to get, right? It's at the also spatial proximity where the tissue region doesn't change that much. Though, those are your neuroanatomy hawks will probably see that even across a couple hundred microns, brain regions like thalamic nuclei will show changes. So, in our case, really, I think it was that match. I want to be able to really map them as closely as possible. The bits that I didn't really show you guys is that I think depending on what reference you look at, you can actually get pretty good results. So, in our preprint, we actually take the Allen Institute, single cell references. They don't represent all different parts of the mouse brain in our Visium experiment here. They are most densely representing different parts of the cortex. We could take them and we could actually really nicely map them into into the Visium spatial transcriptomic data. So I think with organs that have been single celled to death, especially with model organisms, you will get pretty good mapping. But I completely agree that with a lot of human tissues, even though the lymph node stuff that I showed you here was also completely externally generated to our Visium data set, with a lot of human tissues, you're going to run into some of the problems where the reference itself will not match your spatial transcriptomic data sets. If I bore you with one more comment on it, a piece of benchmarking that I didn't show you guys that is actually also related to the reason why we can match fine subtypes is that we also think that cell to location, the way we have modeled it, is also good at taking from the single cell reference what is really supposed to be in the spatial data set. So if you actually had a single cell reference that say has, you know, we actually have this very same benchmark. If you have a single cell reference that have, say, cortical cells versus hippocampal cells, and your spatial data only has cortical, you know, visium spots in it, we show that cell to location is very nice and only mapping the right cell types, cortical cell types into that data set. So we don't really try to overfit. But yeah, I think, you know, moving forward, as we generate more of these data sets, there will be really lots and lots of challenges, both from both the single cell and spatial side.
Hi, Omer.
Hi, Ruben.
Awesome
Hi.
talk.
Thank you.
I was curious, and I agree that, you know, I think batch integration, and especially as these experiments scale across labs and across species, trying to make sense of all this together is going to be a huge computational issue. I'm wondering if you have any plans or can comment on possibly leveraging histological information in terms of batch correction or combination of different data sets. I know that's more of a computer vision problem, but I have a sense that that can even allow for imputation or integration between different types of spatial transcriptomics modalities, like seqfish combined with Visium, for example. I feel like it's seqfish data.
Yeah, I think, Ruben, I think that's a great comment. I agree. I mean, I think you know, we've made sort of like the kind of the simplest use of that histology information in cell to location. So, you know, I like glossed over that a little bit. With cell to location, we actually, a bit more uniquely than other methods, we actually end up estimating basically the absolute cell counts of each reference cell type in each tissue location. So our output is actually really, I think that there are 6 of these cell types in this tissue region. And, you know, that is calibrated with basically tissue level priors, where we actually look at, say, a mouse brain histology image, and we look at the size of a Visium spot, and we could then sort of guess, okay, on average across most tissue locations, you have something like 5 to 10 mouse brain cells, right? And we feed that as a prior and we get this output. So we actually showed that our estimates really nicely correlates. The total number of cells that we estimate for a mouse brain tissue location nicely correlates with this. And I agree that I think that could actually be, even if you're not doing any fancy computer vision stuff, I think that could be a really nice batch integration point, because really you have, even if you have different cells that have different amounts of RNA, you still kind of have, right, like this common point of there are really going to be X many cells based on the histology image. I think moving forward to more complex representations of, you know, histology, computer vision integration onto this, that is, yeah, truly, I think, one of the biggest next frontiers.
Awesome, thank you.
Thank you. Are there any questions?
I have a question. Hi.
Sure. Hi, Dan.
For the spatial transcriptomics, I see that's mostly being used as sort of like a anchoring system, as you will, for the single nuclei. Are there any, I guess, value from the gene expression data from the spatial transcriptomics other than the using it for anchoring? Are there biases that we observe in spatial?
Yeah, absolutely. I think, Dan, that's a very insightful comment. I think, you know, there are a couple different ways you can go about it, right? So there are already a number of good technologies which just look at spatial transcriptomic data alone and pull basically spatially distinct cell type, spatially distinct gene expression patterns. You know, methods like spatial DE or sepal. And, you know, some of those do correlate with regional cell type patterns, others don't. You're correct in that what we are doing here is really using spatial transcriptomics as an anchor to map out these cell types. But I really think that the sort of the next generation of these methods, some of which we are already seeing, such as this really new exciting method that was released from the Nir Yosef's group, they call it DEST, the sort of spatial transcriptomic deconvolution tool of SCVI. They actually have their eyes on also modeling the sort of continuous variation in spatial transcriptomic data, meaning that once you actually map some of these cell types onto the tissue, you can also ask, do these cell types, are they actually showing spatial or tissue microenvironment related gene expression changes in the spatial transcriptomic data beyond what I have in the single nuclei reference? And I am really confident that that will be the sort of the prize for the next set of methods to be able to do that. I think there will be certain limitations based on the spatial transcriptomic data type that you're working with. I think none of the spatial transcriptomic, or if we just sort of talk about sequencing-based technologies for a sec, I think none of the methods have the transcriptomic depth of suspension nuclei yet, though there's also the question of, are we sequencing these spatial transcriptomic data sets deeply enough? I think that hasn't been answered in my opinion. I think there might be a wall in what we can actually probe from these type of spatial transcriptomic experiments. And it might be true that, at some level, you're really going to have to do, like a 10, 20,000 plex in situ hybridization type of experiment to be able to get at these microenvironment related changes.
I have a question for variation correction. So when you talk about batch variation, you assume those samples are very similar and you can correct for batch effects. But actually there might be an interhuman variation, like their gender, their age, and the exact place you cut from more anterior or more posterior. So is there some genes or some library size variation you expect to be standard across all human or all mouse?
Yeah, that's another great question. Thank you. I think the type of batch correction that we're thinking, which we haven't finalized yet, would basically, what I can tell you now is that it would maintain that basically inter-tissue section or inter-human or like a biological replicate heterogeneity that would still be there. We can sort of see that things like library size and complexity are playing a role in things. That's when we generally sort of with subtle location, if you're doing an experiment, let's just say that has as you see in this mouse brain, in this experiment we had something like 5 visium tissue sections across slightly different anatomical levels of the mouse brain. We are actually mapping mapping our single nuclei reference to all of the sections at once. So the batch correction that we are doing really tries to preserve some of that heterogeneity. If we were mapping to multiple people, we would basically pool all of them and map at the same time. Yeah, it's a bit too early for me to say, but I'm hoping that this basically true variation in the spatial transcriptomic data will still be maintained.
Cool.
Hi, Omer. I'm Marco Mendoza. Actually, I wanted to ask you a particular question concerning this particular astrocyte, rare astrocyte population that you found by doing the analysis.
That's right.
Actually, I was actually wondering whether, because you compare different methods, right, for doing the identification for these deconvolution strategies.
Correct.
Right?
Yeah.
And actually, you showed that your methodology is actually basically making better than the others. But my question was, did you try just to use a spatial transcriptome on its own to see whether you can catch such a signature?
Yeah, so
you get
yeah, that's a yeah.
I'm saying so because because recently we have used contiguous JEC cells or pixels, if you want, or these spots in a tool that we published, in which by looking at the continuity of the patterns of the let's say the continuity of the points that could give us a signature would be a nice way in the spatial transcriptome to actually reveal signatures.
Yeah.
So my question is, could we see that already without single cell? That's actually the point.
Oh, yes, a good point. So I think I'd love to check out your method. I'll certainly look it up after this. So I think, in the case of astrocytes, right? So if you looked at basically spatial transcriptomic data alone here, I think what it will tell you is pretty much more or less this. It will basically tell you that there are these like distinct, gene expression patterns across different parts of the tissue. And I think that's driven, you already know in the mouse brain, that's driven by like these regionally enriched cell types. Without single cell data, what I think would be difficult would be trying to basically in this sort of bulk Visium, 55 micron Visium data, try to sort of dissect the signature of neuronal cell types that are known to be localized to the habenula versus regional astrocyte subtypes that we identified to be localized to the habenula. I think that would be a tricky thing. And, I think if your guy's method deals with it, I'd be very happy to look at it. But I expect that it will be difficult to make some of those distinctions without a single cell reference.
Okay, thanks.
Any other questions? As we are waiting, sure, I can maybe ask one question. The lymph node and spleen data, it was very interesting to see the journal centers like lighting up. So you know that the receptors, BCR and VDJ combination, right, how it kind of plays a role in that diversity. Can you guys tap into that question using this, or?
Yeah, I think you could, yeah. So, it depends on, I think, where you get the BCR, TCR information, right? So if you think that you want to get that from the single cell reference, if you have that sequence there, that's mapped here. However, the sort of vanilla version of the Visium assay that comes out of the box doesn't give you BCR, TCR. So you have to keep into account that what you're mapping from, you're actually doing the mapping based on the shared sort of non-TB cell receptor transcriptome, not that. So what you could end up with, basically a bunch of sequences of those receptors that are enriched on here, but you're not going to get that super fine resolution. What I think 10x probably has in mind, because it's really not that complicated, is to add their sort of TCR extension to Visium, right? So you can, on top of capturing the transcripts with a poly A, you can get in there. And I actually expect, I think the FFPE compatible version of Visium, I think it's released next week or something globally. And I think, the technology has been completely revamped. They now use sort of nanostring type of probes that hybridize the transcripts. Then those probes are captured onto the, the sequencing chips. I think that technology, it's trivial to add probes against, T cells, BCRs, whatever in that regard. Yeah.
Right, that will be exciting.
Yeah.
So as we are waiting, the other question is actually about your preference of technology. I see that your postdoc worked on, mostly has spent time on the RNAscope technology, which is imaging based, right?
Right.
And you looked at, many genes, and you looked at this brain in your Nature, I think, neuroscience paper. Then now in your group, I see that you're transitioning into more like nanostring and spatial transcriptomics. So what do you think about this transition? Is it like a, more like tactical transition, or is it practical? Like, what do you suggest to the community?
Yeah, I think you can say that I've been sangarized a little bit in my transition. I think I was certainly, when I came here, I was just like a basically imaging junkie. And, the Sanger Institute is basically a genomic sequencing institute, but, we had this sort of ambition to, go into basically high throughput spatial genomics. I think in my regard, in our research program, I think what we've been captivated with is basically the potential of doing unbiased analyses, right? So that you can actually think about how taking these methods, applying to different tissues and trying to map as much as you can, not just few sort of like select subtypes. And I think that's the kind of, if I go back to really the first slide, I think if you're at the beginning of your kind of single cell spatial genomics journey, I think it really does make sense to invest in sort of these discovery-based, unbiased omics methods, right? To really get the bird's eye view of what's going on. But as I showed with, like, I think astrocytes, what generally happens is that you end up with a number of increasingly better and more confident predictions on where cell types are on different tissues. And you still have to kind of come in at the end of the day with your good old RNA scope and validate what those predictions are. So I think, you know, in our group, I think I do see us investing a lot more still in, you know, unbiased spatial transcriptomic technologies. We are quite interested in, you know, doing high multiplex RNA imaging, part of the work I didn't get to talk about today, where I think it's a very exciting space. And I think, you know, we will still have, I do think that we will still have the biological questions in the group or applications where, you know, that good old 3-plex, 4-plex RNA scope will be the fix, and it will give you that right signal to noise ratio to image that really poor human tissue section. But yeah, definitely more higher plex unbiased stuff for us in the future.
Great, thank you for the insights. Are there any other questions from the audience? If not, let's thank the speaker again. I'm going to use this clapping sign again. Thank you. It was a great talk.
Thanks, everyone.
And before we wrap up, I'd like to also announce that next week we will have Dr. Jeffrey Moffitt from Boston Children's Hospital and Harvard Medical School talking about mapping the transcriptome with NERFISH. And with this, we are concluding today. And thank you guys again for attending. And thank you again, Omer, for this great presentation. And please, all of you, enjoy your weekend and have a nice day for now. Bye.
Thank you so much. Take care. Çok teşekkürler Ahmet. Çok memnun oldum tanıştığımıza.
Ben memnun oldum.
İleride görüşmek üzere. Hoşça kal.
Bye bye.